All right, so let's get started here on our discussion about cardiac output. Finish up our discussion on the heart. So here we have cardiac output, and we mentioned in the previous recording that cardiac output refers to the volume of blood. And I just have to get this out of the way. Ejected by a ventricle each minute. So we went through this in the previous Zoom, but we're going to go through it again to make sure we understand the values and how we can apply those to understanding heart function. So we said there was an equation that we could summarize cardiac output as a combination of heart rate times stroke volume. And stroke volume was the volume of blood ejected from a ventricle each beat or each cycle that we described in the previous Zoom video as well. So a typical stroke volume is around 70 milliliters. And in our example that we used last week, a typical heart rate is around 75 beats a minute. And that combination gives us about 5,200 milliliters per minute. So total blood volume equals pretty close to total blood volume. So all we're saying there is that we recognize that at rest around 75 beats a minute, a good healthy heart will eject 70 milliliters of blood per beat. And over a minute, that represents just about the total blood volume in a typical human being. So it represents, again, another type of efficiency. When we can pump out the total blood volume every minute, it means that we're replenishing oxygen up at the lungs pretty much 100% with each minute as it is able to circulate through the lungs, pick up new oxygen, and get rid of old carbon dioxide on every single cycle. Over a minute, it represents sort of replenishing. Okay, Kazra, stop asking about allowing recording. Okay, it is being
All right, that
recorded.
was my mistake. Sorry. Then
Okay, no problem. It's just that it keeps on flashing up in front of me. So we are recording, so it's all good. Okay, so
sorry, Professor.
yeah, no problem, no problem. It just sort of throws me off a little bit, so I got to get reorganized. And we're recording, so it's a little bit of a waste of your time when you have to listen to all this stuff. Okay, so here we are pushing through total volume of blood every minute. The heart is a very efficient pump in this respect. And really, remember we said last week that the pump is two parallel pumps. The heart is two parallel pumps. So the same thing is happening on the right side as it is on the left. From a clinical perspective, we're much more interested in what's going on the left side because it's the left pump that pumps to the systemic circulation, which means all the cells and tissues in the body that are receiving oxygen for their various functions. So that's really what you'll see from a clinical perspective. This cardiac output is a measure of what's going on the left side, but the reality is it's also happening on the right. You can't get the stroke volume on the left side of the heart if we didn't have good stroke volume on the right side of the heart. So if we looked at one side of the heart, we'll just designate it as blue because it's got deoxygenated blood, and there's our atrium, and there's our ventricle. Okay, it's receiving blood from all the upper body, superior vena cava, the lower body, inferior vena cava. The blood is introduced to the atrium and drains to the ventricle. And from there, we're going to pump up to the right and left lungs, right? So we're just doing a quick sort of summary. Here we have our left lung, there's our right lung. So this really represents the blood that's being pumped from the systemic circulation back to the heart to pick up oxygen at the lungs. So we would breathe in fresh air and oxygenate, add oxygen to the blood that was picked up, that was delivered through the lungs, and it's going to come back and enter the left side. Okay, so the oxygenated blood now enters into the left atrium and gets sent to the left ventricle, and the left ventricle is then going to pump through the aorta to all parts of the body. So we'll fill this aorta in a little bit so that it doesn't look too scribbly, but by now you should be fairly familiar with these anatomical structures that we're drawing in here. And this should just be a quick review again of what's happening. So what we're looking at when we look at the stroke volume is the amount of blood that is loaded into the left ventricle, in this case. And then when the left ventricle contracts, how much of it is delivered to the aorta. So that's our stroke volume, the volume of blood that's ejected from a ventricle. In this case, we're talking about the left ventricle. So the left ventricle, while the heart is filling, remember the cardiac cycle, we said there was a phase, the diastolic phase, which is known as the filling phase, and it's filling up. And then the atrium contracts and puts that final sort of spurt of blood into the ventricle, and the ventricle then contracts. When the ventricle contracts, it delivers a volume of blood, in this case, to the aorta. If we were looking at the right side, it would be to the pulmonary trunk. In either case, that represents our stroke volume. And when we look at cardiac output, it's a measure over time, and we measure the heart rate by minutes. So when we repeat the stroke volume ejection at 75 times a minute, we get our cardiac output. So we should be comfortable with the idea that heart rate is going to be influenced by the autonomic nervous system. We know that the sympathetic division of the autonomic nervous system is fight or flight. It will increase heart rate. And the rest and digest branch, this parasympathetic division, will slow down heart rate. Not just heart rate, but also the force of contraction. Either way, we regulate heart rate through the autonomic nervous system. Now, there are some hormones that can influence heart rate, hormones known as epinephrine, norepinephrine, that are produced from the adrenal gland, which we haven't talked about yet, but that's some other ways of regulating, but our primary way is through the autonomic nervous system. So if you're comfortable with the role that the autonomic nervous system plays on heart rate, you've got that particular factor explained. You can explain it. Now, stroke volume is a little bit trickier because there's different ways to influence stroke volume. So when we look at stroke volume is that final volume of blood that would be in the ventricle before it contracts. So it's got four major factors that you want to be aware of. One of those is called venous return, and we're going to talk more about venous return when we get on the blood vessels in just a few minutes. But that's the amount of blood returning to the heart. In other words, it's the amount of blood that's going to be filling into the ventricle. So we got ways of improving venous return, which would give us more volume, which in effect would give us a better cardiac output. So we'll talk about that. And when we do get a good venous return and blood flows into the ventricle effectively, we create what's called preload. And preload is really referring to the stretch that's put on the ventricular wall. So that equals the stretch on the ventricle wall. So probably the best way to envision that stretch, and I think I mentioned this in last week's Zoom, is think about a balloon. And the more air you put in a balloon, the more stretch you put on the balloon wall, the more force will be exerted when you let that balloon go. And it will fly all over the room until the tension is reduced on the wall of that balloon, and then it'll just drop to the ground. So the same concept applies here. If we stretch the ventricular wall, we create an environment, a situation where we're going to have a more forceful contraction. Now, there's an explanation for that, but it gets a little bit complicated. For now, understand that the more stretch we put on the ventricular wall, the more forceful it will be able to contract. So that will make a better cardiac output. We okay with that so far?
Yes.
Yeah.
Okay. So the third thing that fits hand in hand with what we're talking about is the property called contractility. So with contractility, we're talking about the factors that promote cardiac muscle contraction. So we know a little bit about this because some of it is similar to skeletal muscle. We know that we have to have enough sodium. We know that in the case of cardiac muscle, we also have to have enough calcium, because calcium is an important ion or element that helps to sustain the cardiac muscle contraction. So we would see that in skeletal muscle, when it contracts, it's all or nothing, and it kind of gets drawn out so that it looks a little bit like that. As we're contracting and generating tension, it goes up, and then it resets itself as it comes back down to its base level. A cardiac muscle cell goes up very similarly to the skeletal muscle, but then it has a bit of a plateau before it comes back down. And the plateau is because of calcium. The rise in both cases is from sodium. So we describe the sodium channels in the cardiac muscle cell as fast because they give you that steep curve, and the calcium channels are described as being slow because the calcium comes in a little later but maintains a longer contraction phase. So the cardiac muscle cell sustains its contraction longer, and that's important for the heart. That's important for the heart to be able to generate a strong, longer lasting contraction for this ejection function. So other factors that would come into play would be, so we'll just write in some sodium here and some calcium, and potassium's important too, but also things like acid base and temperature. These would be all factors that would play a role in contractility, making the environment as ideal as possible for a contraction to occur. So these three factors are factors that represent an improved stroke volume. In other words, they'll make stroke volume better if we increase each of these. So if these factors that we've listed and these descriptions that we've listed, the better they are, the better our stroke volume will be. And if our stroke volume is good, then that means our cardiac output is going to be good. So this, for those of you that are maybe a little bit concerned about equations like this one, we're saying that cardiac output is directly related to the combination of heart rate and stroke volume. So when we multiply heart rate by stroke volume, that's how we get our number that we've listed here, just as an example. But if our stroke volume went up to, say, let's just keep it nice and simple, if we made it 100, that would make this number 7,500, cardiac output much larger. And that might reflect, for example, what needs to happen during exercise or some other kind of stress. When there's more demand for oxygen and nutrients in order to make more ATP, we'd expect cardiac output to go up. And if every system in the body is healthy, then we can increase our venous return, we can increase our preload, and we can increase our contractility, all to improve stroke volume. Now, the fourth factor, and I'm going to draw it in a different color, is called afterload. And that is the resistance against the ventricle contraction. So in this case, afterload is a negative. The more resistance there is against the ventricle contracting, the harder the work is going to be. So in effect, if we're trying to increase stroke volume, then we're going to want to decrease afterload. Okay, so we want to increase all three, 1, 2, and 3, but decrease afterload. So I'll show you, if we're talking about that left ventricle, so the left ventricle has a volume of blood in it. Okay, and there's our volume of blood. Okay, and this is our valve that we have to push through, and on the other side of the valve is the aorta. So what we're saying is, if there is lots of pressure here, that represents afterload. So that pressure is going to work against the forces that the ventricle is trying to establish to eject blood. Remember, the blood has got to be ejected into this vessel. So the more pressure working against it on the other side of that valve represents afterload. So if we minimize afterload, we can improve cardiac output. Are you okay with that?
Yes.
Yeah.
Okay, so the simple way to think about that is high blood pressure. We're going to talk about blood pressure next when we get into blood vessels, but I recognize that the higher somebody's blood pressure is, then that represents an afterload on the heart. It means the heart's going to have to work harder. So high blood pressure has a negative effect on cardiac function. And now we can see from this description why that would be. It represents an increase in afterload, and that means the heart has to work harder to overcome that pressure. So in a perfect world, we would like to see increased venous return. That means the blood volume is being returned to the heart efficiently, a good stretch on the ventricular wall. Now, you can have too much stretch, but so we're talking about within reason. And same with venous return and all these factors within reason. They're not never ending, but we're okay with that because we talked about never say never and never say always, except always remember to never say never or always. So this is an example. So we want to maximize our preload, maximize our venous return, maximize contractility, but minimize our afterload. And if we understand that, then we can talk about how stroke volume and heart rate can affect cardiac output, using that equation. So that's where that equation can become an effective way of explaining cardiac function, specifically cardiac output. Okay, any questions about that or anything else associated with the heart?
No questions.
Okay, so we're going to move on to today's topic, blood vessels. So blood vessels are typically, remember we said at the beginning of our discussion on the cardiovascular system that it was a transportation system. It was a system designed to deliver oxygen and nutrients to all the cells in the body and pick up wastes and carbon dioxide from those same cells to ultimately eliminate those things through various mechanisms. We can eliminate the carbon dioxide up at the lungs when we breathe out. So blood vessels are the method of delivery, and the heart was the driver of that delivery. So blood travels in a closed vessel system, which we call our vascular system. Remember, last week we described a pulmonary circulation, which was the responsibility for the right side of the heart, pumped blood into the lungs and back to the left side of the heart. And the left side of the heart was responsible for the systemic circulation, which pumped blood to all the cells and tissues throughout the body. And essentially, the organization goes from very large blood vessels to progressively smaller to the exchange sites, which are called the capillaries, and reversed to return to the heart. So we end up back where we started, our closed circulatory system. So when we leave the heart, it was particularly with the systemic circulation, that large vessel is called the aorta, and the aorta distributes through large arteries to mid-sized arteries to small arteries to what we call arterioles. And from the arterioles, we're going to deliver blood to the capillaries. And capillaries are going to be the site of exchange. And the exchange at the cell level means that we're going to be able to deliver oxygen and nutrients to those cells. And on the other side, pick up waste and carbon dioxide and return that through venules. I'll write all this stuff down. Venules, small veins, mid-sized veins, large veins, back to those vena cava that we mentioned in order to complete the circulation. So we can sort of draw that as aorta, large arteries, small, we'll skip mid just because we don't need to. And the arterioles are small enough, they're going to enter to the capillaries. And from the capillaries, we're going to leave through small veins, large veins, and then the vena cava. And we can stick the heart right in between these two. And complete our circulation. Okay, so we okay with that terminology? We understand kind of what's happening there?
Yes.
Okay, pretty straightforward, I think. So the thing that makes these blood vessels characteristic of their responsibilities are the layers that are created in order to establish these pathways. We don't want them to be leaky until it's important for that feature. And the only place where we really want to see them be leaky is here at the capillaries. So the structure of our blood vessels are based on 3 layers.
Professor, I have a question.
Yeah, go ahead.
I just wrote it on the chat in case if
Well, I don't look at the chat while we're doing the.
oh, sorry. Okay. All right. No, it says you have mentioned earlier when you also giving us a recap for the previous session. I'm always confused with this systemic circulation. right?
Yeah,
That you mentioned in your, is it the entire process? Is it called systemic circulation, or it's only the return part, like when the
no, the entire circulation that leaves the left side of the heart and distributes oxygenated blood to all the cells and tissues in the body and back again is the systemic circulation.
so it will goes to the lung and then back to the
It-
heart.
No, the one that goes to the lungs is called the pulmonary circulation.
Which
The
is
right,
the
the
one that
right
takes
pump,
the
the right pump is responsible for the pulmonary circulation, and the left pump is responsible for the systemic circulation.
okay. So
heart
systemic
is
circulation
two
is the one
parallel
that
pumps.
is good, this the oxygenated blood.
Okay, what happened to your volume? You disappeared.
Okay, so there it means it's two things, right? The systemic and the pulmonary, and one is related with the left one is that comes with the right part, yeah.
Yeah, the right side is responsible for pulmonary, and the left side responsible for systemic.
Okay.
Okay, and remember last
Alright.
week we talked about the fact that the left ventricular wall is much thicker than the right ventricular wall, and this reflects the afterload or the resistance that the left ventricle has to overcome because it has to push blood through the entire systemic circulation. And the right pump only has to pump to the lungs, which are right beside the heart and are more or less at the same level as the heart. So the work of the right pump is much less than the work of the left pump.
Okay, yeah.
Does
Okay.
that make sense?
Yeah, yeah. I'm always just confused with this too. That okay. Thank you, Professor.
Okay, good.
All right.
I'm sure that not just you appreciates that explanation. So let's hope anytime you've got a question, go right ahead and ask. But I don't check the chat until we're on a break. So use your microphone. Feel free to use your microphone as far as that goes. So our structure of a typical blood vessel is going to be described by these three layers. The innermost layer is called a tunica intima. And the tunica intima typically is the lining of the inside of our blood vessel. So the space or the hollow part of a blood vessel is called the lumen. So this tunica intima lines the lumen. It's an epithelial tissue. You remember from PNA 100 that we talked about epithelial tissues having an open end, an open surface. And in this case, it opens into the blood vessel. So this is also sometimes described as an endothelial lining or layer. And it's typically one squamous cell layer thick. So that was called a simple squamous layer or a simple squamous tissue. And it rests on a basement membrane. They're packed close together to form a nice smooth surface so the blood is able to travel down the length of that blood vessel without too much interference. The next layer, the middle layer, is called the tunica media for middle, and it's mostly going to be made up of smooth muscle. So you might remember smooth muscle was under involuntary control. It has some capacity to contract against a stretch, but also will be regulated by the sympathetic division of the autonomic nervous system. Okay. Probably you discussed during fight or flight scenarios in the autonomic nervous system discussion that most blood vessels do not have any innervation or supply from the autonomic division. They have a supply from the parasympathetic division. They have a supply only from the sympathetic division. So the sympathetic division of the autonomic nervous system can cause both vasoconstriction and vasodilation to blood vessels. Now, that should sound a little bit confusing to most of you, because how is it that the same neuron can cause both a dilation or a constriction? Does anybody have any ideas on how that might be explained?
No.
So if you remember, and I hope you do, because
Probably
the-
we might have some neurons that have mixed function, motor and sensory function.
okay, no, we're not talking about motor and sensory here. We're talking about motor only. The sympathetic and the parasympathetic divisions of the autonomic nervous system, they're a motor, which means they're going to land on effectors, and effectors fall into the category of muscle tissue, either skeletal, cardiac, or smooth, or glands. Those are your big effectors. And the only one that the sympathetic would not have a direct effect over would be skeletal muscle, because that's voluntary, and the autonomic nervous system is subconscious. So what we're talking about here is the sympathetic nervous system signals its targets with epinephrine, in other words, the neurotransmitter that is going to target a receptor on smooth muscle or cardiac muscle is epinephrine, or its close cousin, norepinephrine. And we can use those. We don't have to differentiate for our purposes, either or. Okay, so it's the same messenger. The point I'm making here is, in the case of the sympathetic nervous system, it can target some blood vessels and cause dilation, and other blood vessels and cause constriction. And we know generally which is which, because if we recall fight or flight, we want to increase blood flow to tissues and cells that support fight or flight, but decrease blood flow to tissues that do not support fight or flight. So, for example, we're going to decrease blood flow to the digestive system and to the kidneys, because they are not helpful in our flight or fight mechanism. But we want to increase flow, for example, to skeletal muscles and to the brain to help us with fight or flight. So if we're using the same messenger, but getting different effects, we have to change the receptor. Okay, so now generally speaking, we have what are called alpha receptors and beta receptors that are sensitive to epinephrine or norepinephrine. Now, typically, and this is a big summary, this is a big generalization, when we match epinephrine with an alpha receptor, we're going to get vasoconstriction, reduced flow. So, for example, we've got alpha receptors on the smooth muscle of the digestive tract. So when epinephrine is released during fight or flight, it targets the alpha receptors in the digestive system and causes that blood flow to be reduced. So it slows down activity in the digestive system. And the beta receptors, they're going to promote vasodilation. So they're, for example, to the heart and to skeletal muscle. So we want those tissues to be improved upon during fight or flight. Does that make sense? You okay with that?
Yeah, but I'm wondering if epinephrine increases the blood flow or reduces the blood flow.
That's exactly what I just finished telling you. If you have vasoconstriction in a blood vessel, is that going to increase flow or decrease flow?
Or increase the flow, constriction?
No, vasoconstriction.
Constriction.
You're constricting the diameter of the blood vessel.
It reduces the blood flow.
It will reduce blood flow. So that's what I've been saying. Okay, you got to, maybe what I have to do there is just listen slowly instead of sort of panicking a little bit. When you target an alpha receptor that results in vasoconstriction, you're reducing the diameter of the blood vessel, which means you're going to reduce blood flow to that group of cells. For example, the digestive system. When the beta cells meet up with epinephrine or norepinephrine, vasodilation is the result, and that will increase the diameter of the blood vessel, which means more blood to locations like the heart and the skeletal muscle, which fits with what we understand about our fight or flight response. We want the heart rate to go up. We want the skeletal muscles to be contracting because we have to decide whether we're going to fight or flight. Okay, this is why what I said last week, it's extremely important that you remember what you learned in AMP 100 about the autonomic nervous system in this case, right? Fight or flight versus rest and digest. So that's the innermost layer, tunica intima. The middle layer is a muscular layer. And our third layer is called the tunica externa. E-X-T externa. So that's the outermost layer of the blood vessel. And that's the layer that's going to give that blood vessel a little bit of protection. It's going to be a more tough, fibrous type of connective tissue. It's going to be the location for where some of our sensory receptors are for blood pressure that we'll be talking about a little bit later. But that's the other layer that's typically part of the organization of our blood vessel. So if we looked at the outside of a blood vessel, and that was an artery that we wanted to look at, and actually let's make it a little bigger. So let's make our artery, diameter of our artery a little bigger, and the diameter of our vein about the same. Okay, so our outermost layer, the one that's red for the artery and blue for the vein, would be our tunica externa, and it's made up of some fibrous connective tissue. But the first thing you're going to notice is that if we looked at the internal diameter of an artery versus the internal diameter of a vein, we would see that the walls are much thinner in a vein. And if we understand form and function, there's a reason for that. So what do you think the reason might be for why the walls of an artery are much thicker than the walls of a vein?
Probably the arteries may carry the large volume of blood.
So say that again?
As it might be probably the arteries can be carrying large volume of blood than the veins.
So you think because of the smaller diameter of the blood vessel, it'll carry more blood?
Yep.
How would you figure that?
The bigger
It's
the
a
diameter, the larger the volume. That's what I'm assuming.
Nope, I don't think your logic kind of follows based on what you've said. What we have here is a blood vessel that's under much greater pressure. Arteries are under much greater pressure. Remember where they come from. The arteries are leaving the heart. So here's our pump. And we leave the heart through the aorta. The greatest amount of pressure in the systemic circulation is going to be in that aorta. And as the aorta branches off, the pressures are going to continually drop. So we can draw a schematic of that, and we will in just a minute. I'll show you what that looks like. But the point I want to make is, if we looked at the outside diameter of two blood vessels, one being a vein, one being an artery, we would see that the arterial wall is much thicker. it represents a functional difference. Structure and function are related. So when we look at a thicker wall, just like we looked at the thicker ventricle, it was because there was going to be a force that required resistance against that pressure. So the arterial wall is under much greater pressure. It's going to be thicker, and it reflects that purpose. Veins are under relatively low pressure, and they're going to need another way of delivering blood back to the heart, which we're going to talk about when we look at the organization of a vein in just a second. But when we look at the distribution of pressure, let's go back here. So here's our structure of a blood vessel, just like we were talking about. So here's an artery, and here's a vein, okay? And we can see that the thickness of the media compared to a vein is greater. So that diameter there is greater than this diameter here. The outside thickness is also greater, reflecting the increased pressure that an arterial blood vessel is going to be under compared to a vein. And capillaries, we haven't talked about the capillary yet, and we will be talking quite a bit about the capillary, but it's a simple squamous epithelium, very, very thin, and is going to favor exchange, which we'll talk about in a few more minutes. Okay, now the other feature to be aware of in sort of mid-sized to larger veins is that they have valves. And valves, we already know from talking about the valves in the heart, they're designed to encourage one-way flow. We want to encourage one-way blood flow, returning blood towards the heart in this case. So these flaps are going to be open when the blood is flowing towards the heart and designed to close when the blood is trying to move back away from the heart. So we'll talk about how that's a possibility in a couple of minutes as well. We okay with the structure there, those three layers?
Yes.
Okay. Now, I'm not sure how big this picture shows up for you, but here, what I want you to look at is just a little schematic of what's happening to the pressure as we move from the greatest pressure at the aorta to the lowest pressure in the veins. So it's just a schematic. This is just showing us an upper and lower limits, homeostasis. So we could even, you could even draw that in as being represented by fluctuations as we move through upper and lower limits. But the concept is that on average, we may see a pressure plus or minus around 100 millimeters of mercury as it leaves the aorta. And as we distribute down through from large arteries to small arteries to arterioles, the pressure is progressively being decreased. Till we get into capillaries, capillaries are described as being relatively low pressure exchange vessels, and we'll explain how they're exchange vessels when we get into our description of those capillaries. But the idea is large arteries to capillaries, and then from capillaries, small veins to big veins to the vena cava when we return back to the heart. But you can see that the pressure on the venous side is quite low. So we describe the aorta and large arteries. They're sometimes described as being elastic or conductive vessels. So they're responsible for moving the blood from, in this case, from the heart down into the body, systemic circulation. So the large arteries are not just, there's no exchange going on here. Then we progress to mid and small arteries. And these ones are considered to be the distribution or distributive arteries. So they're less about the elasticity and conduction and more about distributing. So, for example, there's an artery called the brachial artery that's going to supply the arm. There's a femoral artery that supplies the leg, femur, femoral. There's a trunk that goes to the stomach and the intestine. So those would be described as distributive. And then the smallest of the arteries are called arterioles. And we mentioned that these arterioles are the ones that are going to deliver blood to the capillaries. And then from the capillaries, we're going to empty into small veins called venules. And these venules are going to go into small veins, larger veins, to the vena cava. And again, in between the two, we've got the heart. So the heart delivers to the aorta and the vena cava return to the heart. So this represents our systemic circulation from progressively smaller arteries under greater pressure. There's our pressure. And as we get down to capillaries, much, much lower pressure, and exchange is going to happen there. So we put in our exchange. And then the rest of the blood has to be returned to the heart. What did we call that? That was called venous return. And of the total blood volume in the body, at rest, there's a total of more blood in the veins than in the arteries. And that's because the veins are under relatively low pressure. So it's a bit slower to get the blood back to the heart. You can appreciate that when someone's standing upright, not only are the veins under lower pressure, but they are also moving against gravity. They have to go uphill. Think about pumping the blood from your foot all the way back up to your heart, right? So we have to have some mechanisms that are going to help us with this venous return. We don't need that one just yet. We don't need that one just yet. Let's go back up. So veins are under low pressure and often work against gravity. So we need some help. We need some ways. The pump that delivers to all oxygen and nutrients to the whole body is generated from the heart. But once we get down to those capillaries, we don't have a second pump down there to help us return. So we're dependent on some other mechanisms. So one of those factors is called a skeletal muscle pump. So what that means is we've got a vein, and on either side of that vein, we have muscle. So think about muscles in your legs. Okay, and the veins that are in your legs are going to pass between two or more of these skeletal muscles. And when the muscle contracts, it gets shorter and fatter. So what it's going to do when it gets shorter and fatter is it's going to squeeze the vein. And when it squeezes the vein, it's going to push the blood. Now, in theory, it could push it in either location. But what prevents it from going backwards that we mentioned already?
The valves.
Right. We've got our one-way valves. So if I make this vein just a little bit bigger so we can see the valves a little easier, the valves are designed to be open when we push blood back towards the heart and fall closed if we try to push blood back away from the heart. And that direction, that push, will come from skeletal muscle pumps. So what we learned, for example, in factories is that if you had your workers standing at a station for too long in one position, their blood started to pool in their legs and they had to sit down or they got tired and they had to take a break. But if you had someone work at a workstation for 5 or 10 or 15 minutes and then move to another workstation, that moving created skeletal muscle pumps. It helped return blood back to the heart. Your worker didn't get as tired, and it's understanding this mechanism of activating skeletal muscle pumps and understanding that one-way valves will ensure the blood is pushed back towards the heart and not away from it. Now, there's a third factor that comes into play here. This one's a little more abstract, but it's called the intrathoracic pump. So our thoracic cavity is where the lungs and the heart are located. So remember when we drew the heart, remember we said last week it's about a fist shaped, sits on the bottom of the diaphragm there, and the lungs that are inside this thoracic cavity they sit in here, and when you do the respiratory system, we'll be doing it, we'll understand that for the lungs to stay inflated, we have to have negative pressure inside that thoracic cavity, and that helps keep the lungs inflated. So this negative pressure helps to create a bit of a vacuum or a draw or a pull. So we have a blood vessel that's outside the thoracic cavity, and we're trying to return it to the inside of the thoracic cavity. When we're under negative pressure compared to positive pressure, we're creating a draw, like a vacuum. We're creating a pull. And so the blood vessel inside that thoracic cavity is able to expand in that negative pressure and create a vacuum or a pull. Does that make sense? Do we understand that?
Yeah.
Okay. Now, that's not a huge factor, but it is still a factor encouraging venous return. So when we think about venous return, there's these three factors that we want to be able to discuss or talk about that improve venous return. Healthy skeletal muscles, moving. The reason why you don't want to sit or stand in one place for too long is that you lose the effect of these skeletal muscle pumps. The one-way valves, probably you've seen what happens sometimes when one-way valves stop working properly. The blood will begin to pool in those veins. If you look at, might be a grandma or your mom, she might have some veins that look kind of, they're close to the surface typically they're going to be found in the legs. And I mention a grandma or a mom because it's a little more common in women than it is in men, but it happens in both, where you can see that the veins look a little bit bluish and there'll be these sort of stretched out segments in the vein. They look a little bit bulbous. And those are where one-way valves are not effectively pumping or ensuring that the blood is moving towards the heart, and they're pooling instead at the location of that valve and putting a stretch on the blood vessel. So the blood vessel might look a little bit like this, where the blood is pooling up at the location of the closed valve. And if that's allowed to progress, then eventually that valve becomes faulty. And when the valve becomes faulty, it's going to stop ensuring one-way blood flow, and instead it's going to escape and start to pool up. And that's where, grandma might have swollen ankles at the end of a day. And that's a reflection of these valves that aren't doing their job as effectively as they once did. So that's venous return. That's the important function that we talked about. We mentioned venous return when we talked about cardiac output. This is where it comes from. This is what we're talking about when we talk about this venous return. Okay, now let's look a little closer at our capillary network and what needs to happen at our capillary network. So when we go from an aorta that's under the highest pressure and distribute to arteries, and then from arteries to the arterioles, the smallest of the arteries, okay, we have what's called a terminal arteriole. The terminal arteriole finishes on the capillaries. So it would look like this. Here's our terminal arteriole, okay, and it came from branches that started at the aorta. And it's going to finish in a capillary network. So the capillary network is a series of branched, simple squamous epithelial capillaries that are going to be located where we find cells. And the other side of that capillary network, we're going to leave through a venule. So this would be a venule. This would be, whoops, I'll do it in red, an arteriole. So we're delivering blood and we're taking it away. And in between this capillary network are cells. Okay, so everybody knows what we've drawn here. Down at the cellular level, we have this capillary network that surrounds the cells and sets up an exchange site. We okay with that so far?
Yes.
Okay. Now, on the arterial side, we're under relatively high, what's called hydrostatic pressure, water pressure. So because we're in a capillary, we call it a capillary hydrostatic pressure. And on the venule side, it's a low capillary hydrostatic pressure. Okay, now on this side, the high hydrostatic pressure, high water pressure encourages filtration. So when we get into these leaky segments here, it's going to encourage filtration. In other words, net pressure outward. So that's going to encourage filtration. And where we have low hydrostatic pressure, it's going to encourage reabsorption. So the yellow arrows represent filtration, and the blue arrows represented reabsorption. So you have to understand that when we want exchange to occur, we need to create a driver. And the driver is going to come from this hydrostatic pressure. But we don't want to lose everything in the capillary network, or we won't have any volume to return back to the heart. Everybody with me on that one so far?
Yes.
Yeah.
Okay, so our hydrostatic pressure solves the problem of filtration, delivery, but it doesn't explain reabsorption. We need another force. So the other force is called osmotic pressure. The other force is the osmotic pressure that's created from solute that does not get exchanged. So your liver makes these things called plasma proteins that you should have learned about when you did blood in PNA 100. But the plasma proteins are too big to be filtered, so they create osmotic pressure along with whole cells, mostly red blood cells. So what we get is a relationship where we have high, we got high capillary hydrostatic pressure minus what we're going to call our blood colloid osmotic pressure, and that's going to equal filtration. And our low capillary hydrostatic pressure minus our is going to give us reabsorption. Okay, now you can see by the arrows that I drew up here, we've got reabsorption going back to the capillaries, and we've got filtration leaving the capillaries. So filtration is going to deliver, and reabsorption is going to return. So we're going to put some nice simple numbers to this to help explain what's going on here. This should be an L in there, filtration. So we're going to make our, and these are reasonably accurate numbers, but I've simplified them a little bit just so we can watch what's happening. These numbers are not numbers you need to memorize, but just understand the concept. So if our blood colloid osmotic pressure, all of those initials, B for blood, colloid means it didn't leave. It's a solid that's creating this osmotic pressure. So, excuse me. We're going to give our blood colloid osmotic pressure the number 25, and it doesn't change throughout the length of this capillary. It's once the colloid osmotic pressure is established, it's going to be the same all the way through. Okay. Now, if our capillary hydrostatic pressure we said was higher on the arterial side, so we're going to make that number 35. So our net filtration pressure is going to be equal to what?
What's to 10?
10.
It's 10, right? The hydrostatic pressure is higher. So we can even, and this will make more sense in a minute, we'll give it a plus 10. Now, our capillary hydrostatic pressure on the venous side, on the venual side, okay, it's much less. It's been knocked in half because we've lost a lot of the water and other factors that increase pressure on the arterial side through filtration. So this number is going to be around 17. And when we subtract 17 from 25, our reabsorption is going to be what number?
-8.
-8.
Right.
-8.
-8. So what we're saying is reabsorption, we're going to designate it with a negative number, giving it the opposite direction. So it's going back in, or reabsorption is going to be represented by a negative number, right? And filtration by a positive number. Does that
Then
make
what this one-
sense?
That means the just a drop. The absorption dropped.
Yeah, as our blood pressure goes through the capillary network, it continues to drop. So, and it dropped about a half from 35 to 17. And that makes it less than the osmotic pressure. So now the osmotic pressure becomes a draw. In the interest of equilibrium, the water that was pushed out during filtration is going to be reabsorbed in the second half. So in the interest of homeostasis, we're trying to reestablish blood volume. But look, we got a bit of a problem. Those two numbers are not equal.
Yes.
So that means what? That means there's a partial loss of total blood volume, right? Right?
Yes, there's a loss. Negative
There's
2.
going to be a net loss. So we have to have a mechanism, again, thinking about the principle of homeostasis, there must be a mechanism available to us to pick up the difference, to pick up the extra that is not being picked up during normal exchange, like what we've described here. So the difference, the lost blood volume, is picked up by lymphatic vessels. You'll learn about the lymphatic vessels when you do the lymphatic system and the immune system. But that's the picture that I had showing you. Let's go back to... So let's look at this picture first. Okay, this is showing you these pressures. Okay, so here's filtration. So we know that we're going to use 35 and 25 for our osmotic pressure. And that's going to give us a net of positive 10. And a positive number is describing filtration. So that's net pressure outward. Filtration is outward. At the opposite end, where we're returning blood volume to the capillary, we've got well, they use a -7, but we can make it an 8 to be consistent with what we talked about. So we'll make that a 7 and that a 25, and that gives us a -8. And because we put a negative sign on it, it represents reabsorption. So we're returning. But you can see there's a difference. +10 out, -8 back in. So we're going to have a lymph vessel that's nearby. This lymph vessel is close by, and it picks up the difference and returns it to the circulatory system. More about that when you do the lymphatic system, but that's what this picture I was showing you before. Here's our lymphatic vessel. So it's kind of in the middle of the capillaries, and all these beigey colors are cells. So we filtered out, reabsorbed in, and the difference is going to be picked up by this green vessel, the lymphatic vessel. So if we wanted to kind of show this from a capillary point of view, here was our arteriole, and it branched. We'll simplify it. The second half of the capillary network. Like that, and then to understand where the lymphatic vessels would be, the lymphatic vessels would be running in between. And they would pick up the extra. And then our cells would be all in this area here, nearby, so that they could receive the nutrients and unload the waste. Does that make sense?
Yes.
I'm saying, can you go over it again?
So our filtration pressure was +10. Reabsorption pressure was -8. Those two are not equal. We have a problem. The lymphatics vessels look after the difference. In fact, let's take that terminology out. They pick up the difference. Okay, so you can see here, we're really talking about 20% of volume, right, is lost if we just looked at the capillaries themselves, the blood capillaries, right? We got a 20% differential there. So we have to have a mechanism that reestablishes the volume, and that's the responsibility of these lymphatic vessels that we find at all of our capillary sites. So they're going to pick up the difference. Does that make sense? Yeah, we're okay with that?
Yes.
Yes.
Okay. All right, so now let's continue on with our capillary exchange. So keep in mind that the purpose of this circulatory system is to deliver oxygen and nutrients to all the cells in the body so that they can first make enough ATP to function and then utilize some of the other nutrients for other structures. Maybe they're going to synthesize a hormone, or maybe they're going to repair a cell membrane and they need some amino acids and so on for that sort of thing. So the purpose of capillary exchange is delivery of oxygen and nutrients and pickup of waste and carbon dioxide. So we want our capillaries to be leaky. They're relatively low pressure and leaky. So we have three types of capillaries, three main types of capillaries. One type, the most common type, are called continuous capillaries. So what they would look like is the capillary vessel itself is a simple squamous epithelium with a basement membrane. And the basement membrane is just almost like think of it almost like a cheesecloth or something that's partially porous, but is going to provide some structure to the simple squamous epithelium. So if we looked at the simple squamous epithelium, remember they're flat and single layered, and periodically along a simple squamous epithelium in a capillary, there's a little gap. So this gap allows exchange to occur. Anything that is small enough to fit through that gap is going to be able to pass from high pressure to low pressure that we already talked about when we talked about that capillary pressure. So if it's filtration, it's going to pass outward. If it's reabsorption, it can come back. And the other thing is that these are a squamous cell. So squamous cell means the distance across the cell is relatively short. So if we were looking at the blood vessel on the other side, we would have these tight junctions and every so often a bit of a gap. There's another tight junction like that, and then maybe another gap. I'm exaggerating the gap. But the idea here is when the pressure is high, then things are going to move out. When the pressure is low, then things can move in. Reabsorption, filtration. Okay, now that's one type. That's called a continuous. B is a type it's called a fenestrated capillary. Fancy word. And fenestrated capillaries are set up much the same as this one we've just shown you. So we would have a cell that's flat and connected to the next cell. And then every so often there would be a gap and another gap. Okay. But the other thing is that this fenestrated capillary also has holes in its membrane. So that makes this cell much more leaky. So if we looked at this cell membrane like this, we would see that there are holes. So not only is the blood vessel got some gap junctions associated with it, but the cells themselves are also pretty porous. So that makes movement inside and outside this capillary even easier. So more leaky. You follow me okay with that?
Yeah.
And then the most leaky are called sinusoidal capillaries. So with sinusoidal capillaries, they've got everything that we've already talked about, in addition to the basement membrane also being leaky and the spaces between the cells being much, much larger. So I've got a picture to show you of that will help explain this as well. Here we go. All right, so our first one is our continuous capillary. You can hardly read that, I'm sure, but the idea is we've got a capillary with a single cell layer thick with spaces in between that are going to allow for exchange. So that's typical. That's what we see in typical groups of cells. A fenestrated capillary, the next one, has these holes in the membrane in addition to those gap junctions. Makes it even more leaky. And then our most leaky are called sinusoidal. So we've got large fenestrations, large gaps. That's where the word sinusoidal comes from. And even the basement membrane is incomplete. So this is continuous. This one is a discontinuous. So it's the most leaky of the bunch. So you're only going to find those in very, very specific locations. There's a few places where, for example, in the liver, where this type of sinusoidal capillary is important. Places where we would see the middle group, where we want more exchange than what we see in a typical situation, would be, for example, in the digestive system, where we want to be able to absorb nutrients in the digestive system to bring them in from our stomach or our small intestine. So different ways that make exchange more or less easy based on the structure. Those are structural changes. Okay, now, from a functional point of view, capillary exchange is going to be driven so we said structurally, we had our continuous, fenestrated, or sinusoidal. And from a functional point of view, we got lipid soluble substances. So anything that's lipid soluble is going to be able to move across the plasma membrane from an area of high pressure to an area of low pressure. So examples of structures or substances that are lipid soluble are gases, oxygen and carbon dioxide. So they're going to move across a plasma membrane, cell membrane, simple squamous epithelium, just because there's an area of high concentration to an area of low. So the arterial blood has high oxygen, and the cells have been using oxygen, they're low. So we're going to be able to move from inside the blood vessel to out because we have that gradient. And the CO2 is being produced in the cell, and as a result is relatively high, while it's also low in the blood vessel. So we move CO2 from the working cell back into the blood vessel. Does that make sense? So if this was our oxygenated blood vessel, and then it became a vein, okay, on the oxygenated side, it's very high in O2, and it's low in O2 over here, and we're low in CO2 in the oxygenated blood. So this is our direction. We're going in this direction. And here's our busy cell. So there's the nucleus of the cell, and there is mitochondria. So the oxygen, because this cell has been making ATP, it's low in oxygen. And as it makes ATP, it creates CO2. So CO2 is high and it's low in the blood. So we're going to move in that direction. And O2 is high and low. So we're going to move in that direction. That's how we're going to get lipid soluble molecules to move from an area of high concentration to an area of low. Remember, lipid soluble means they could cross the plasma membrane. They don't need a channel or a leaky pathway. They're going to be able to cross the plasma membrane. We okay with that?
Yes.
Okay. Now you should also remember that cells are capable of endo or exocytosis. So some products that are being produced inside a cell will be able to be delivered through exocytosis from inside the cell to out, or picked up from outside the cell in endocytosis. For example, some hormones will be delivered in this mechanism, this way. If they are larger proteins or hormones, they may have to utilize endocytosis or exocytosis. If they are able to pass through those clefts, those spaces that I talked about, if they're small enough to pass between cells, between the endothelial cells, they'll go from high to low. So that's the way some electrolytes like sodium and potassium can move that way. Glucose can move this way. Small proteins or amino acids can move that way. And then the fourth mechanism that we can take advantage of is by adding the structural component that we talked about before through fenestrated and or sinusoids for larger molecules. So we got four mechanisms that we can take advantage of for exchange. All of them will be influenced by that bulk flow that we talked about, the hydrostatic pressure being greater at the arterial end that encourages filtration, and being less at the venous end that encourages reabsorption. Okay, if you can't move, if molecules can't cross through 1, 2, 3, or 4, then they're going to remain inside the capillary. And that's exactly what happens to plasma proteins and whole cells, like red blood cells and white blood cells. So those are what create the osmotic pressure. The blood colloid osmotic pressure is created mostly from these plasma proteins, but additionally from those cells. So that explains the way that we exchange at a capillary. They're relatively low pressure, leaky vessels that favor movement based on 1, 2, 3, and 4 that we've listed here, and based on the pressures that generate filtration versus the pressures that generate reabsorption. Okay, we're going to take a break.